The naive idea was to try to use blood contamination of human mouth fluid as a non-invasive approach for serological analysis in old and very old person. Old and very old person, of course, do not like to have a mass biopsy, and also blood sampling, and therefore to find something different could be very important. Though considered a problem, blood contamination of saliva may offer interesting opportunities for the follow-up of frail old person submitted to rehabilitation strategies. Saliva sampling may be performed more frequently than blood sampling, nothing to say muscle biopsies. Our first goal was to validate a simple, cheap methods to measure blood contamination of saliva and eventually non-traumatic methods to increase its blood serum content. Literature presents preliminary data that toothbrushing may have a significant effect on the protein composition of saliva, in particular by contamination with serum constituents. Therefore, before analyzing myokine in saliva of frail old person, in this very preliminary study, we started the process to confirm or not the previous results. 7 volunteers participated in the study. All volunteers were over the age of 65 years and reported to have no systemic diseases. They were instructed to abstain from smoking, eating, drinking caffeine-containing beverages, and toothbrushing at least 1 hour prior to the test. First, unstimulated whole saliva was collected for 5 minutes by simple drawing in a test tube. Next, the volunteers brushed their teeth for 2 minutes using their personal toothbrushing without toothpaste. Saliva was then collected for 5 minutes by drawing in Eppendorf vials, which were centrifuged at low speed for 5 minutes, and then the supernatant stored at -80 degrees until use. Lysozyme, fibrinogen, and serum albumin concentrations in the supernatant, improperly diluted samples of saliva and of a pool of control human sera were measured by ELISA method according to the instruction and manufacturing using ABCAM tests. Our early results that were based mainly on studying serum albumin and lysozyme suggested that toothbrushing, those kind of toothbrushing, may increase the blood contamination. But when we optimize saliva dilutions and added to the analysis fibrinogen into saliva, collected from saliva before and after brushing, all the saliva had measurable amount of analytes, including the fibrinogen, which is a minor component of the pooled sera. Sera is what remains after coagulation, and therefore the vast amount of fibrinogen disappear and remain just traces. By this analysis, we did not recognize any statistically significant difference in analyte contents, suggesting that the measurable blood contamination is a frequent event in elderly person, even without a very low trauma. We think that the analysis of fibrinogen is a promising reference to quantify serological biomarkers through a non-invasive procedure, because you have exactly something that remains after blood coagulation that is high, of course, in the blood sampling, and relatively low, but not so low to become measurable. Here you see that before and after brushstatin, that amount of brushstatin, there are no statistical difference. Notice that there are changes if you use linear interpolation or logarithmic interpolation, because you have to compare high concentration with very low concentration, and to have in the same test to save money, it is much better to use this trick. Then we decided that we had enough evidence that some blood contamination is present in, let me say, normal saliva. We moved to the analysis of two myokines, let me say, not those secreted by the salivary gland, but those that may represent the systemic response in the subjects, because they are related to the amount of blood measured by the trick of fibrinogen. TNF, in our case, is the pro-inflammatory myokine or cytokine. Interleukin 10 is a well-known anti-inflammatory myokine. We were able to measure the Barbara and Sandra, and not me, of course, were able to measure the amount of the two myokines, and with the trick to use the ratio between the pro and anti-inflammatory myokines, we believe that we are really measuring what is present in the saliva as in the consequence of the blood contamination, because in 6 of the 7 different subjects, the ratio between TNF-alpha and interleukin 10 is near 1, which is the expected value for normal serum, and indeed it is the same that we have in our test condition when we analyze the pool serum. In conclusion of this very preliminary, but in my opinion relevant demonstration, TNF-alpha and interleukin 10 concentration in properly diluted sample of saliva and of a pool of control human sera could be measured by using the ELISA method according to the instruction of the manufacturer, using the ABCAM ELISA kit. Our preliminary results show that human saliva of 7 normal over 65-year person, the content of fibrinogen is almost 1/10 of that present in a pool of sera, matched healthy seniors, and indeed it is a measurable common amount. All the human saliva, all the 7 human saliva contain measurable amount of TNF-alpha and interleukin 10. The ratio was very similar to that described in literature and measured in our control pool of sera. In conclusion, fibrinogen is a promising reference to quantify serological biomarkers through a non-invasive procedure and by a relatively low-cost approaches. Its determination will increase acceptability and frequency of myokine analysis during follow-up in aging and rehabilitation. Thanks to all the patient audience.
Sorry, Helmut Kern from Vienna, but you don't see me. I have problems with the connection.
Doesn't matter. It is important that you.
How much was your estimation of the content of TNF alpha in the saliva? Because fibrinogen is only 1/10, you said, in comparison to blood. And how much is TNF alpha?
You see here.
Yeah.
The pool of sera, the fibrinogen, please note that the scale are different. Here are very, very low amount and then abruptly go up.
It's a logarithmic scale.
In the logarithmic scale, there is no, I have not here this is not myokines. This is only fibrinogen.
Yeah, this 1/10.
It is the marker of the presence of serum in the saliva.
Yeah. Okay.
And these go to 30,000. This is around 5,000 or less.
Okay.
And therefore it is around 1/10.
Yes. And how 10%. And how much is the concentration of TNF alpha in comparison to blood?
Again, the pool serum is about 100.
Yeah.
The saliva are here 5, 6, 4, 3, and therefore it is 5% of the pool serum. You agree?
Yeah. If you said fibrinogen is about 10% and TNF alpha is 5% of the level of the serum.
Yes, and the same is true for the other myokines. Both myokines have more or less the same amount.
Thank you very much, Ugo. It's a fantastic talk, and congratulations to your energy of the last 2 years, and to Barbara. Thanks to Barbara to find all this.
Padova, Sandra, and Giovanna.
Yes, of course.
And my contribution was only to oblige them to do a stupid experiment, but I am a lucky man. Maybe we have something worth to continue.
Okay, thank you very much, Ugo.